Okay. Hi everyone. This is me, K. Lakshmipati, APRJC Namakur. Welcome back. Regarding this topic, digestion and absorption. Regarding this chapter, so far we have completed the discussion about the introduction as well as the alimentary canal or gastrointestinal tract or simply gut. What are the parts involved in the gut and what are the functions of each and every, what are the responsibilities of each and every part of the gut and what is the histology, all these things so far we discussed. Particularly in this class, I am going to deal with another subtopic, that is digestive glands. Digestive glands which will helpful in the process of digestion. Before we are going to understand the process of digestion, first of all, we should know about the different kinds of digestive glands related to our gut. Related to our gut. To understand properly the process of digestion, first of all, we should know how many types of digestive glands, what are the secretions from each and every digestive gland, what is the role of each and every digestive juice which will secretes from the digestive glands respectively. So broadly we have many digestive glands. First of all, broadly we can divide them into two types. Broadly we can divide them into two types. First one is glands associated with the gut. Glands associated with the gut. The second one is glands in the walls of the gut. Broadly, they can be classified into two categories. First one is glands associated with the gut. That means some glands, for example, salivary glands, liver and pancreas. Salivary glands, liver and pancreas. These glands are very closely associated with the gut. They will pour their secretions into the lumen of the gut. They are very closely associated with the gut. Salivary glands, liver and pancreas. Whereas the second type of glands in the walls of gut, walls of the gut. That means they are completely embedded in the walls of the gut. They are completely embedded in the walls of the gut. They themselves, the glands themselves, embedded in the walls of the gut. Examples, gastric glands and intestinal glands. These glands completely embedded in the walls of the gut. So let us discuss one after the other. First one, glands associated with the gut. Regarding this, first of all, let us take salivary glands. Salivary glands. Salivary glands. Salivary glands. How many kinds of salivary glands that we have? What are they? Where they are located? What is the nature of its secretion, what is the role of the secretion in the process of digestion, all these things which we are to be discussed. Salivary glands. There are three pairs of salivary glands in our mouth cavity. In our mouth cavity, there are three pairs of salivary glands in our mouth cavity or buccal chamber or oral cavity. Oral cavity. There are three pairs of salivary glands. What are they? One pair of parotid glands, one pair of parotid glands, and one pair of submaxillary glands. These submaxillary glands are also known as submandibular glands. Submaxillary glands are also known as submandibular glands. The third pair of glands are called sublingual glands. One pair of parotid glands, one pair of submaxillary glands or submandibular glands, or one pair of sublingual glands. Three pairs of salivary glands which are located in our buccal chamber. Where they are located, each and every gland, where they are located. For example, parotid glands are located in the cheek, below the pinna, at the base of the pinna. One pair of parotid glands located in the cheek at the base of the pinna. Pinna is the external part of the external ear, pinna. Here these parotid glands are located. And submaxillary glands or submandibular glands. These are present or these are located at the angles of lower jaw. First pair of salivary glands are called parotid glands, which are located in the cheek, below the pinna, at the base of the pinna, both sides, one pair. Similarly, submaxillary glands or submandibular glands at the angles of lower jaw. And third pair, sublingual glands. These glands are present below the tongue. Whatever the secretion secreted by these glands is called saliva. Whatever the secretion that is secreted by salivary glands is called saliva. When we see in each salivary cells, each salivary gland is made with two different types of cells. Serous cells and mucus cells. There are two different types of cells. by which each salivary gland is made up of. These cells collectively secrete saliva. These cells collectively secrete the secretion which is called saliva. It is slightly acidic, with a pH value of 6.8. Saliva is slightly acidic in its chemical nature with a pH value of 6.8. Saliva comprises with more water, mucus, along with water and mucus, It has some electrolytes. Such as chloride, bicarbonate, sodium, and potassium. Saliva slightly acidic in nature with a pH value of 6.8. What are the components of saliva? Water, mucus, more water and mucus, electrolytes such as chloride, bicarbonates, sodium and potassium. Along with these water, mucus, electrolytes, saliva is provided with the enzyme ptyalin and lysozyme. There are two enzymes in saliva. One is ptyalin. Ptyalin is a salivary amylase which act on carbohydrates, starch, or we can say they are polysaccharides. Ptyalin is also known as salivary amylase, which act on carbohydrates, polysaccharides, or starch. Ptyalin convert the polysaccharides into disaccharides, such as maltose. Salivary amylase or ptyalin. This is the role of ptyalin. Amylase or ptyalin act on polysaccharides, and those polysaccharides are converted to disaccharides such as maltose by the action of the salivary amylase ptyalin. And what about this lysozyme? Lysozyme is a bactericidal in nature. Lysozyme is a bactericidal in nature. Bactericidal. That means it will kill harmful bacteria that enter into our mouth cavity along with the food and water. It is bactericidal enzyme which can kill harmful bacteria that will enter into our buccal chamber along with the food and water. Harmful bacteria are killed by the action of this bactericidal enzyme that is lysozyme. That is lysozyme. Okay, this is about the salivary glands. There are three pairs of salivary glands, one pair of parotid glands, one pair of submaxillary glands, and one pair of sublingual glands. Whatever the secretion secreted by these glands is called simply saliva. If you take each and every gland, salivary gland, each gland is made up of two types of different cells, serous cells and mucous cells. These cells collectively secrete a slightly acidified juice that is called saliva with a pH value of 6.8. The components of, when we see the components of saliva is comprises with more water, mucus and electrolytes such as chloride, bicarbonates, sodium and potassium. Along with these, saliva is provided with two important enzymes. One enzyme is ptyalin. Second one is lysozyme. Ptyalin is a salivary amylase which can act on polysaccharides and they will be converted into disaccharides such as maltose by the action of salivary amylase, that is TAM. And second enzyme, what is the role of lysozyme? What is the role of lysozyme? It is bactericidal enzyme, bactericidal in nature. The harmful bacteria which enter into our buccal chamber along with the food and water, those will be killed by the action of this lysozyme. This is the significance of saliva and about salivary glands. Next, let us take another gland which is very closely associated with the alimentary canal, our alimentary canal, that is liver. That is liver. Liver is a largest gland in our body. Liver is a largest gland in our body. Largest gland. The largest gland in the human body is liver. It is located the right side of our abdominal cavity, just below the diaphragm. It is located right side of the abdominal cavity, just below the diaphragm. It is the largest gland. It is considered as a master gland. It is considered as a master gland because other than digestive function, liver has, other than digestive function, liver has so many functions. It participates in detoxification and it participates in carbohydrate metabolism, fat metabolism, lipid metabolism. There are so many functions are performed by the liver. Most important, a vital organ in our body, the largest gland. It is also considered as a master gland in our body. It is located in the abdominal cavity, right side of the abdominal cavity, just below the diaphragm. It has two lobes, right and left lobes. Right and left lobes. It has two lobes. For example, if you take a picture of liver, it has two lobes. This is a right lobe and this is a left lobe. Below the lobes, we can see a pear-like, a pear-shaped or oval-shaped sac-like structure. Below the lobes, at the center of the lobes, two lobes, below the liver, we can see a bulb-like, a sac-like, oval or pear-shaped structure. That structure is called gallbladder. That structure is called gallbladder. That structure is called gallbladder. Gallbladder. Each lobe is provided with so many lobules. Each lobe is provided with like this, so many lobules. For example, each lobule, for example, this is one lobule. Each lobule is surrounded by a connective tissue layer. This is a connective tissue layer. This is the lobule, one lobule of liver. One lobule of liver. Each lobule is surrounded by a connective tissue layer. That layer is called Glissen's sheath. Glissen's sheath. Each lobule is covered with or surrounded by a connective tissue layer. That layer is called Glissen's sheath or Glissen's layer. When we see the inside the lobule there are we can see several hepatic cells. This is hepatic cell. Inside the lobule, these hepatic cells are arranged in a hepatic cells are arranged in a cord-like manner. So this is called hepatic cord. Hepatic cord means the arrangement of cells in a serial manner. So many cells are arranged in a cord-like structure. That cord is called hepatic cord. Each lobe of liver consists so many round lobules. In each lobule, hepatic cells are arranged in a cord-like manner. That is called hepatic cord, hepatic cells. The lobule is covered or surrounded by a connective tissue layer. That layer is called Glissen's layer, Glissen's sheath. Okay. Hepatic cells secretes a juice called bile. Bile juice. Hepatic cells secrete a juice called bile juice. A small duct is given by each and every hepatic cell. A small duct is given by each and every hepatic cell. That is called hepatic. That is called hepatic duct. Hepatic duct is given by hepatic cell. Each hepatic cell has a small duct that is called hepatic duct. All the hepatic ducts will join together and form a common hepatic duct. A common hepatic duct. Each hepatic cell have a small duct that is called hepatic duct. All the small hepatic ducts will join together and forms a common hepatic duct. This common hepatic duct will join with another specific duct that is called cystic duct. Cystic duct. The common hepatic duct will join with a specific duct which is called cystic duct. Cystic duct is given by gallbladder. So, for example, this is a common hepatic duct. This is common hepatic duct will join with a cystic duct which is given by the gallbladder and forms common bile duct. A common bile duct is formed by the union of common hepatic duct and cystic duct which is given by gallbladder. This common bile duct extends and will join with a pancreatic duct, this is pancreatic duct which is given by another digestive gland that is pancreas and forms hepatopancreatic duct. Hepatopancreatic duct is formed by the union of common bile duct and pancreatic duct. The common bile duct is formed by the union of common hepatic duct and cystic duct which is given by gallbladder. So this hepatopancreatic duct will open into the duodenum. This is duodenum. This hepatopancreatic duct opens into a duodenum, which is the first part of the small intestine. At this opening, in between hepatopancreatic duct opens into duodenum. At this opening, that opening is regulated by a sphincter called sphincter of Oddi. Sphincter of Oddi. So both bile juice and pancreatic juice will enter the duodenum. Both juice will be poured into the lumen of duodenum. At the opening of this hepatopancreatic duct into the duodenum, the flow of juice is regulated by a sphincter. That opening is guarded by a sphincter. That sphincter is called sphincter of Oddi. Sphincter of Oddi that regulates the flow of juice into both the bile and pancreatic juice into the duodenum. And what is the significance of this gallbladder? What is the role of this gallbladder? There are two functions for gallbladder. One is storage of bile. Storage of bile and making it in concentration. To concentrate the bile is the first function of the gallbladder, and storage of the bile. Whenever it is necessary, the contraction of this bulb-like structure gallbladder, some amount of bile will pass through the common bile duct into the duodenum. So temporarily it is the storage organ of bile, and the bile is concentrated in the gallbladder. These are the functions of gallbladder. This is the significance of gallbladder. And what about bile? We are discussing about the largest gland, the vital gland, an important gland, master gland of human body, liver. It has so many functions other than digestion. It plays vital role in so many metabolic biochemical processes. Bile. Bile. It has no enzymes, no specific enzymes in the bile, though it can play vital role in the process of digestion. How? Let us discuss. It is alkaline in nature. The pH value of 7 to 8, more than 7. It is alkaline in nature, more than 7. It is alkaline in its chemical nature with a pH value of more than 7 or 7 to 8. No enzymes. What are the components in the bile? Bile pigments, and bile salts. Bile pigments, bilirubin, biliverdin, urochrome, these are bile pigments which can be excreted through our excretory system. Bile pigments, there is no use of bile pigments. Bile salts, there are two types of bile salts, sodium and potassium. Sodium and potassium glycolates. Sodium and potassium glycolates and tarocholates. Sodium tarocholate, sodium glycolate, potassium tarocholate as well as potassium glycolate. So sodium and potassium glycolates and tarocholates. Bile salts, they have a significant role in emulsification of fats. Actually, fats are the larger molecules than carbohydrates and proteins. The enzyme which can act on fats is called lipase. The enzyme which can act on fats is called lipase. So lipase will not able to act on fats directly because they are in the form of larger molecules. So first of all, these fat larger molecules are to be converted into smaller ones or micelles. Fats are converted into micelles, smaller molecules of fats. So the conversion of large molecules of fats into small molecules or micelles of fats by the action of bile salts is called emulsification of fats. Then only the lipase from pancreatic juice or from intestinal juice can act on emulsified fats only. So the lipase can act on emulsified fats only. They could not act on fats directly unless the emulsification of fats takes place. So the process of digestion, particularly the process of lipid digestion, the emulsification of fats is very, very, very important process. For that process, these bile salts, sodium and potassium glycolates and sodium and potassium taurocholates will help. They are converted into larger molecules of fats are converted into the smaller globules. Those are called micelles. Then only the lipase can act on fats. This is the significance of emulsification of fats. The role of bile salts. Okay, there is no specific role, there is no specific enzymes in bile, though it can play a significant role in the process of digestion, particularly in the fat digestion. Okay, this is about the liver. This is about the liver. Okay, in your textbook also, there is an important topic, that is the functions of liver. Digestive function. Just now I discussed about the function related to digestion, the function of liver by secreting bile, which is comprised with bile salts, the role of bile. This is the digestive function. Other than digestive function, liver has so many functions in our body. It is a vital organ in our body, as it has so many functions. Liver is considered as a master gland. Liver is considered as master gland. Let us discuss about the functions of liver, and then we will go to discuss about the the second largest gland, that is pancreas. We will discuss later. First of all, now I am going to deal with the functions of liver other than digestive function. What is the digestive function of liver? It secretes bile. Bile has bile salts along with bile pigments. Bile salts participates in the emulsification of fats. Then only the lipase enzyme can act on emulsified fats. That is the significant role of liver regarding to the digestive function. Other than digestive function, as far as carbohydrate metabolism, liver has so many functions. First one is glycogenesis. Glycogenesis. What does it mean? What is meant by glycogenesis? What is meant by glycogenesis? Synthesis of glycogen from sugars. Synthesis of glycogen. What is glycogen? The excess of sugar in our blood is being carried to the liver, where by the influence of a hormone called glucagon, insulin, that sugar converted to glycogen, and it is stored in the liver. Glycogenesis takes place in liver. The synthesis of glycogen, simply the synthesis of glycogen from sugars is called glycogenesis, which occur in a liver. That glycogen is stored in the liver. That glycogen is we can say it is a animal starch. Animal starch. By process photosynthesis, the end product is actually glucose. During the daytime, that glucose is converted to starch, which is not soluble in water. During night, that starch will again converted to glucose in the form of glucose solution. It is transported to various places of the various parts of the plant. That starch is the plant product. And similarly, a glycogen is animal starch. Glycogenesis. It is the responsibility of liver. And then glycogenolysis. Whenever it is necessary, the glycogen which is stored in the liver is broken down into glucose. That is called glycogenolysis. Lysis. That means breakdown. Whenever it is necessary, this glycogen will break down into glucose. That process is also occur in liver. So glycogenolysis. And then gluconeogenesis. Gluconeogenesis means it is a synthesis of glucose from synthesis of sugars from non-carbohydrate molecules. Synthesis of glucose or sugars from non-carbohydrate molecules such as lactate, pyruvate. These are non-carbohydrate molecules. The synthesis of the production of glucose or sugars from non-carbohydrate molecules such as pyruvate, lactate. The process is called gluconeogenesis. It also occurs in the liver. Next, lipogenesis. Lipogenesis is the process by which lipids, fats and triglycerides the formation of the synthesis of lipids or fats and triglycerides from glucose and other substances, that is called lipogenesis. It is also the responsibility of liver. And then the role of lipid metabolism. Lipid metabolism. The role of liver in lipid metabolism. There are two responsibilities. One is synthesis of cholesterol and production of triglycerides. Synthesis of cholesterol and synthesis of triglycerides. These two functions are also concerned to liver. And protein metabolism. Regarding this, deamination of proteins. Deamination of proteins. Deamination. What is meant by deamination? The removal of amino group from a molecule. The removal of amino group, NHA group, the removal of amino from excess amino acids participate in an ornithine cycle in the liver. In this process, from the excess amino acids, the amino group is removed. The amino group is removed from a molecule. Amino group is converted to ammonia. Later, that ammonia will convert into urea through ornithine cycle. That ornithine cycle, the chemical reactions of ornithine cycle is also responsible for a liver. The excess amino acids are undergone a process called deamination. Simply, what is deamination? The removal of amino group from a molecule. The removal of amino group from a molecule is called deamination. That removed amino group will convert into ammonia. Later, that ammonia will convert into urea. That urea will enter into the blood circulation, and it is supplied to our kidneys. Automatically, that urea is removed through a process called excretion, elimination of nitrogenous wastes. So, deamination process. And then Cori cycle. Cori and Cori cycle. During anaerobic muscle contraction, you have already learned that different types of respiration, aerobic respiration and anaerobic respiration in the previous classes. I think that is in 10th class. During anaerobic respiration, muscle contraction, lactic acid, an intermediate product, that is lactic acid is formed. Because of the accumulation of this lactic acid in the muscles, we will have muscle fatigue. Because of the accumulation of this lactic acid, we will have fatigue. So, whenever it is necessary, sometimes our muscles participate in anaerobic respiration. Because of that, an intermediary substrate is formed, product is formed, that is called lactic acid. Because of the accumulation of this lactic acid, we may have muscle fatigue also. Okay. So such lactic acid is converted to glycogen. That lactic acid is converted to glycogen through a process called gluconeogenesis. Glycogen. And sugars are produced from other than the carbohydrate substance, that is lactic acid, through chorecycling, which takes place in liver. This is another function of liver. And then detoxification. Detoxification of toxic substances. Liver is the first and most important check post in our body. Liver is the first and most important check post. It detoxifies. Detoxification means a removal of harmful toxic substances, neutralizing the toxins or harmful substances which enter into our body along with food and water before they enter into the blood stream. They enter into the bloodstream in the liver. The harmful substances are detoxified by the hepatic cells. The SER, smooth endoplasmic reticulum of hepatic cells, play a key role in detoxification. Some toxins, poisonous substances, harmful substances that enter into our gut along with food and water, before they reach into the blood circulation, they are detoxified by the liver. Hepatic cells, particularly a cell organelle, smooth endoplasmic reticulum of hepatic cells, will play a key role in detoxification of poisonous substances. Next, liver also act as a thermoregulatory organ. Thermogenesis. As it has more glucose reserves. It can also act as a thermoregulatory organ by producing heat whenever it is necessary. So through a process called thermogenesis. So liver is also act as a thermoregulatory organ. And then act as a hemopoietic organ. Liver can act as a hemopoietic organ in infants, not infants, in the fetus. In fetus, liver act as a hemopoietic organ. The production of RBC. Hemopoiesis means the production of RBC. So liver will also act as a hemopoietic organ. The new RBC will form in the liver, particularly in a fetus. Whereas uh, in adults, it act as hemolytic organ in adults. In adults, aged, dead, worn out, damaged, pathogenic RBC split some number of RBC can destroy in the liver. So liver can also act as hemolytic organ, particularly in adults. Hemolytic organ or hemolytic or erythroclastic organ in adults. Whereas in fetus, liver act as a hemopoietic organ. It produces new RBC. Next, synthesis of plasma proteins. Synthesis of plasma proteins is also one of the responsibilities of liver. Plasma proteins. There are different kinds of plasma proteins. Albumins, globulins, fibrinogen, prothrombin, heparin. These are plasma protein. Liver synthesizes all these plasma proteins. Particularly, fibrinogen and prothrombin are blood clotting factors which help in the process of blood coagulation. Whereas heparin is an anticoagulant. It prevents the clotting of blood in normal conditions while blood flows in blood vessels. So that is the normal condition. In such a way, it prevents the coagulation or clotting or agglutination of RBC. This is anticoagulant, and these two are blood clotting whenever it is necessary. When we are wounded and when the blood vessels are ruptured, when blood oozes out, then it is needed. The process of coagulation is needed. At that time, particularly these two proteins will play a major role in the process of coagulation. Next, liver has Kupffer cells. Liver has Kupffer cells. Kupffer cells. What are Kupffer cells and what is the role of these cells? Kupffer cells are in between the hepatic cords, sinusoids, hepatic cords, in between the hepatic cords. What is hepatic cord? The arrangement of cells in a rod-like structure. Serially, they are arranged one after the other. The total arrangement of that hepatic cells is called in the form of a cord, that is called a hepatic cord. In between two hepatic cords, sinusoids, canals like small canals like structure sinusoids. In sinusoids, the macro, these are large phagocytic cells that remove unwanted substances and microbes. Kupffer cells are large phagocytic cells. Kupffer cells are large phagocytic cells which are present in the sinusoids of the lobules of liver. In each lobule, there are so many hepatic cords. In between hepatic cords, small canal-like structures. These are called sinusoids. In these sinusoids, these large phagocytic cells are present. These are called Kupfer's cells, which play in removal of unwanted substances and microbes by by a process called phagocytosis. They These cells themselves phagocyte engulfed all those unwanted substances along with some harmful microbes. It is a protective function of liver. Hence, we already discussed that a liver is the first and most important check post in our body, as we discussed in this class. Just okay, and then liver detoxifies alcohol to some extent. Liver detoxifies alcohol to some extent before it reaches into the blood circulation. So, liver is the first and most affected organ in chronic alcoholism, cirrhosis of liver, chronic alcoholism, chronic alcoholism, also called cirrhosis of liver. Liver is the first and most important affected organ in chronic alcoholism or cirrhosis of liver. So it detoxifies. Liver detoxifies alcohol to some extent before it enters into the blood circulation. Next, ketogenesis. What is ketogenesis? It is a catabolic pathway. Ketogenesis is nothing but a catabolic pathway ketogenesis is the catabolic pathway through which some energy is provided. The mitochondria of hepatic cells, in the mitochondria of hepatic cells, the ketogenesis takes place. Fatty acids and some ketogenic amino acids are converted into ketone bodies in the mitochondria of hepatic cells. That process is called ketogenesis, through which some amount of energy is released, so that it is a catabolic pathway. These are the functions of liver. Okay, with this we completed two digestive glands. First one is salivary glands. Second one is liver. We discussed about the liver. First we discussed about the liver, its digestive function, the role of bile in the process of digestion, and then so far we have discussed the functions which are not related to the digestive function of liver. It has so many functions. Moreover, apart from this, it considered as a master gland. That is the reason it is considered as a master gland. Okay, with this we completed two digestive glands. First one is salivary glands and second one is liver. The functions of liver also discussed. It is a very important topic for your IPDs. In the next class, the remaining digestive glands, pancreas and other the glands which are completely embedded, glands themselves completely embedded in the walls of gut. They are gastric glands and intestinal glands will be discussed in the next class. Thank you. Okay.